Hello my dear friends, welcome to Live Prep. In the previous lecture, we studied about the nucleotides with structures, names, everything we have completed. And now in this lecture, we are going with the nucleic acids. What are these nucleic acids? DNA and RNA are called as nucleic acids. And here there is a small information. In the NCERT, they have not given with this much of clarity, but for our understanding purpose, we will go with some clarity. Okay. And then we will see what are the functions of this DNA and RNA. With that, this lecture will be completed. And here observe carefully, in the previous lecture we studied about nucleotides. Nucleotides are what? They are the monomers of nucleic acids. If we see the insoluble fraction, acid insoluble fraction, acid insoluble pool or acid insoluble fraction, it is also called as retentate. In this retentate total we are getting 4 components. What are these 4 components? Already we have studied about proteins, and now in this lecture we are studying about nucleic acids. Nucleic acids. In the next coming lectures we will study about both polysaccharides and lipids. Polysaccharides and lipids. Lipids. Yes or no? Out of these 4, only these 3 are true macromolecules. Why these 3 are called as true macromolecules? These are the actual polymers. These are polymers, whereas lipids are not polymers. These are not polymers. Okay. Anyhow, in the next lecture we will study about lipids only. So in this lecture we will study about nucleic acids. Already we studied about the proteins. Proteins are polymers of amino acids. Nucleic acids are polymers of nucleotides. In the previous lecture we studied about individual nucleotide. Yes or no? So adenylate acid, guanylyl acid, cytidylate acid, uridylic acid, thymidylate acid. With respect to DNA, by adding deoxy we will get all these. But anyhow, in the DNA uracil is not present. In the RNA, thymine is not present. This is the one point that you need to remember. So in this way, proteins already we studied, and now we are focusing on nucleic acids. And nucleic acids are also part of true macromolecules. Yes or no? So with this lecture will be completed. In the next lecture, we will study about lipids. After that, we will go with carbohydrates in general. Actually, carbohydrates information is very huge, so that we will divide into monosaccharide, disaccharides in one lecture and polysaccharides separately in a different lecture. So slowly we will understand all the points with clarity. Okay. Now we are talking about the nucleic acids, which are the true macromolecules, and these are observed in acid insoluble fraction. Clear? This is the basic information. And these nucleic acids are polymers of nucleotides. Suppose this is one nucleotide, this is another nucleotide. Likewise, many nucleotides are attached to each other. And now this entire molecule is called as nucleic acid. This is called as nucleic acid. And nucleic acids are 2 types. Please observe this carefully. Nucleic acids are 2 types. Nucleic acids are 2 types. What are these? What are these? One is called as RNA and other one is called as DNA. And you need to know the full names. What is the full name of RNA? RNA is called as nucleic acid. RNA is ribonucleic acid. Why it is ribo? Because it has ribose sugar. It has ribose sugar. So ribose sugar containing nucleic acid is ribonucleic acid RNA, whereas DNA is deoxyribo. Already we studied about 2 deoxyribose. So that deoxyribose is present. That deoxyribose sugar containing nucleic acid, that's why it is called as deoxyribonucleic acid, DNA. Understood or not? So the abbreviation is DNA. Full form is deoxyribonucleic acid. Abbreviation is RNA. Full form is ribonucleic acid. So in this way, if you clearly see, based on the type of sugar, please remember this point, very important for examination. Based on the type of sugar that is present, nucleic acids are 2 types. Ribose sugar containing nucleic acids are called as ribonucleic acid RNA. Deoxyribose containing nucleic acids are called as deoxyribonucleic acid DNA. So in this way, 2 types of nucleic acids are present. But anyhow, with respect to ribose sugar and with respect to deoxyribose, we studied individual nucleotides. Yes or no? Adenylic acid we studied, deoxyadenylic acid we studied. Guanylic acid, deoxyguanylic acid. Cytidalic acid, deoxycytidalic acid. In case of RNA, uridalic acid is present. In case of DNA, thymidalic acid is present. Yes or no? With one or two examples in the previous lectures, we studied even the structures also. And now here in this lecture, what I will do is, I will tell you how these nucleotides are attached to each other. Even though clear-cut information was not mentioned here, anyhow in the MBI they have mentioned, and here in simple they have given the information. So I will try to make it very simple. Please remember, with more details, I will explain you in the molecular base of inheritance chapter. Here, to understand NCERT lines, you need some basic information. So that much of basic information I will give you. Clear? Now let us see. Here what we need to understand, nucleic acids are polymers. Polymers means what? Many units are attached. First what we will do, if you understand how two units are attached, how two nucleotides are attached, if you understand this, same is the remaining process also. What we studied for proteins. In the proteins, first we studied how two amino acids are attached. Once you understand that reaction, same is for remaining all amino acids also. Same way, here also we are focusing on two nucleic acids. How two nucleic acids are attached to each other. If you understand that, same is the process for remaining nucleotides also. So in this way you can understand how nucleic acids are formed. And please remember, here I am not giving the complete structure. In simple, I will try to close this content. So now let us observe this carefully. First what we need to take, so first we need to take two nucleotides and then we will see how these two are attached. First I will take one nucleotide, this is the first nucleotide, and then I will take second nucleotide. Only two nucleotides I will explain. If you understand this, same is the process for remaining nucleotides also. First nucleotide means one nitrogenous base, one sugar, and then one phosphate should be present. Yes or no? At least these three should be present. So here what we will take, I am not drawing the complete structure, simply I am writing A. A means that is the adenine structure. You know what is the adenine structure. So let us take that adenine structure here. And then here, for understanding purpose, first I will take the deoxyribose sugar, because we are talking about DNA first. I will tell you, simply by changing that, DNA RNA will be changed. Okay. So here what I will do is, I will take deoxyribose. In the deoxyribose, what happens? Here, instead of OH, H is present. Yes or no? And here, OH is present. Here, OH is present, but anyhow, in the nucleotide, nitrogenous base and sugar are attached, so they are attached. Okay. And next one, here it will be CH2O. This is the phosphate, P double bond O, OH, OH. Yes or no? This is the nucleotide. But in the previous lecture itself, I told you, sometimes single phosphate is present, sometimes two phosphates are present, sometimes three phosphates are present. Yes or no? If single phosphate is present, it is called nucleoside monophosphate. If two phosphates are there, nucleoside diphosphate. If three are present, nucleoside triphosphate. In this way, we are giving the names. This information also I told you. But please remember, whether it is for RNA synthesis or DNA synthesis, whatever it is, always three phosphates are required. So that's why here it is not single phosphate, it should be three phosphates. It should be three phosphates. So in this way, the three phosphates are attached to each other. This is how we are drawing this. And now what is the name of this? Adenine plus deoxyribose. It is called deoxyadenosine with its three phosphates. Deoxyadenosine triphosphate. This is what the name of this molecule. And here, since we are talking about DNA, here also we need to take deoxyribose sugar containing. And here for convenience, let us take the cytosine. See, anything you can take. Okay. Here I am taking the second nucleotide cytosine, and this is also attached to deoxyribose. It is also attached to deoxyribose in this way. This is the hydrogen. This is the OH. And here also CH2O P. This time I am writing double bond O on this side and OH is here. O P double bond O OH, O P double bond O OH and OH. So here also This is the second nucleotide. Here also 3 phosphates are present. Cytosine plus deoxyribose, it is called deoxycytidine plus 3 phosphates. Deoxycytidine triphosphate. This is the name of the molecule. So this is one nucleotide, this is another nucleotide. Yes or no? Now let us see. Already in the previous lecture we studied what is this bond. This bond is N-glycosidic bond. N-glycosidic bond between nitrogenous base and sugar, N-glycosidic bond. What is this bond? This is also same bond. It is N-glycosidic bond. It is N-glycosidic bond. Understood or not? Next one. Between the sugar, this is the fifth carbon, right? 5' carbon. Between this sugar and the first phosphate, there is a bond. What is this bond is called as? This is called as a phosphoester bond. Try to remember these names. Phosphoester bond. Between two phosphoric acids, between two acids, it is anhydride bond. So which anhydride bond? It is a phosphoanhydride bond. Phosphoanhydride bond. Anhydride bond. This is also between two phosphoric acids. This is also phosphoanhydride bond. Same way here also, simply I am writing phosphoanhydride bond. PAB means phosphoanhydride bond. This is PAB, phosphoanhydride bond. This is phosphoester bond. PEB I am writing, phosphoester bond. So this is phosphoanhydride bond. This is also phosphoanhydride bond, like this one only. And here between phosphate and sugar, what is this one? Phosphoester bond. This is how normally these nucleoside triphosphates are present. So when two such nucleotides are coming very closely, now for the DNA synthesis, DNA polymerase enzyme is required. If it is RNA, RNA polymerase enzyme is required, but the mechanism is exactly same. Enzyme is different. In the nucleotides here, deoxyribose for DNA and ribose for RNA will come, but the mechanism of attachment exactly same. So that's why if you know for DNA, the same is for the RNA also. So that's why observe carefully. This is one nucleotide, this is another nucleotide. Now what happens? Now observe carefully. For the first nucleotide at the 3' position, OH is present. For the first nucleotide at the 3' position, OH group is present. Now this OH group will interact with OH group of first phosphate. First phosphate is called alpha phosphate. Second is beta. Third one is called as gamma phosphate. So 3' OH group of first nucleotide will interact with OH group of alpha phosphate of second nucleotide. So here there is a reaction. Now what happens? In this particular process, what happens? When it is interacting here, to make a bond it needs energy. So who is broken here? This phosphoanhydride bond is broken. Observe carefully. Once phosphoanhydride bond is broken, it will get its own OH, this will get its own OH. That means this molecule is separated. And what this molecule is called as? Having two phosphates. Having two phosphates, it is called PPI. If it is single phosphoric acid, Pi. But if two phosphates are present, it is PPI. It is PPI. It is called as a pyrophosphate. Pyrophosphate. Two phosphates are present, pyrophosphate. PP, pyrophosphate. But what is this I? I is inorganic. I is inorganic. So now this molecule having two phosphates is called inorganic pyrophosphate, and this again having one anhydride bond, and there is an enzyme called pyrophosphatase. That enzyme will come and break this bond again, and once this bond is broken, now this phosphate is separated, this phosphate is separated, two individual phosphates. So in this way, this is broken into two molecules of Pi, inorganic phosphates. This is one inorganic phosphate, this is another inorganic phosphate. In this way, what is the ultimate conclusion? First nucleotide 3' OH is attacking on alpha phosphate of second nucleotide. During this process, on the second nucleotide, last two phosphates are removed. And when they are removed, what happens? High energy bonds called phosphoanhydride bonds. Both the anhydride bonds are high energy bonds. They are broken. When these bonds are broken, they will release the energy, and that energy is used in making this particular bond. And how the bond will be formed? Observe carefully. OH group of phosphoric acid and hydrogen from 3' OH of first nucleotide. This OH and hydrogen, this is removed in the form of water molecule. So now water molecule is removed. Now let us remove this. Now water molecule is removed here. Observe carefully. Now water molecule is removed. That means here O remains and here this P. And now there is a bond that is formed between these two. This O is directly connected to phosphate of the second one. Now observe carefully. Now first nucleotide is attached to the second nucleotide. Understood or not? In this way the bond is formed. And what is this newly formed bond? Only this bond. Only this bond is called as phosphoester bond. It is not diester, it is single ester. Di means two. So it is single ester only. This is phosphoester bond. This is phosphoester bond. And now you need to observe the concept carefully. Many students are making mistake here. Now this is the 3' carbon. 3' carbon 2. This is the 5' carbon of second one. Between 3' to 5', observe carefully. Newly formed bond is phosphoester bond, but we are talking the bond between two carbons. What is this carbon? 3' carbon of first nucleotide and here 5' carbon of second nucleotide. That's why it is 3' to 5'. What is this bond? For this phosphate, on this side there is one phosphoester bond. On this side there is one phosphoester bond. This is also phosphoester bond. So this is one phosphoester, this is one phosphoester. That means between these two carbons, what is present? Two phosphoester bonds are present. That's why it is called as phospho diester bond. Diester bond. So regarding this bond, in the previous NCRT, in the biomolecules they have mentioned, but in this new NCRT they have deleted this information. But as it is, this information was given once again in molecular basis of inheritance. There once again I will explain you, but here you just understand the basic information at least. So one nucleotide is attached to another nucleotide through 3' 5' phosphodiester bond. This is what they are saying. So in the case of protein, what we are saying, two amino acids are linked by chemically called amide bond. The same is biologically called as peptide bond. So in the protein, two amino acids are attached by amide or peptide bond. But in the case of DNA or in the case of RNA, whatever it is, in the case of DNA or RNA, the two nucleotides are attached by phosphodiester bond. Understood or not? The two nucleotides are connected by phosphodiester. Di means what? Two ester bonds are there. Where? Between 3' of first nucleotide and 5' of second nucleotide. So in this way, this is one phosphoester bond, this is already present ester bond. So two esters are present. That's why it is called a phosphodiester bond. So in this way, two nucleotides are connected by phosphodiester bond. And now for the second nucleotide, once again 3' OH is present. And now this will attack on alpha phosphate of third nucleotide. Same reaction will occur. Once again phosphodiester bond will be formed. In this way, many nucleotides are attached to each other. If only two nucleotides are present, it is called as dinucleotide. If three nucleotides are present, it is called a trinucleotide. In this way, if many are present, polynucleotide. That's why DNA and RNA are called as polynucleotides. Many nucleotides are attached to each other. Understood or not? So in this way, what I will do is, I will keep dot like this. Once we are keeping dot, what is the meaning of this? Many are attached in the same way. And now what is this complete molecule is called as? Now this complete molecule is called as a DNA. Why this complete molecule is called as DNA? Because it is having deoxyribose. That's why it is called DNA. Suppose instead of H, suppose it is having OH. Here also instead of H it is having OH. Then it is called as RNA. Because it is having ribose sugar. That's it. But mechanism of attachment exactly same for both DNA and RNA. So if you know for one, automatically you can understand for the other one. But simple difference is this one only. If hydrogen is present at these locations, it is the DNA. If hydroxyl OH group is present, it is the RNA. Attachment mechanism, everything is exactly same. But for DNA synthesis, DNA polymerase enzyme will come. For RNA synthesis, RNA polymerase enzyme will come. That's it. This is what the information is. Clear? In this way, we are talking about this particular information. Now let us see some important names relation with respect to proteins and nucleic acid. Nucleic acid. In the case of protein, what is the each unit is called as? In the case of protein, each unit is called as amino acid. In the case of nucleic acid, each unit is called as nucleotide. Nucleotide. Please observe this one. In the case of protein, each unit is called as amino acid, but in the nucleic acid, each unit is called as nucleotide. Next one. Next one. What is the bond that is present between two amino acids? It is the amide bond. It is the amide bond, also called as peptide bond. Peptide bond links two adjacent amino acids. But here, what is the bond? It is a phosphodiester bond. It is the phosphodiester bond that is connecting two adjacent nucleotides. This is what they are saying. And here, if two amino acids are present, here if only two amino acids are present, it is called as dipeptide. Here, if only two nucleotides are present, it is called as dinucleotide. Observe this carefully. If three amino acids are present, it is called as tripeptide. Here also, if three are present, it is called as trinucleotide. Names almost same, but here peptide, here nucleotide names we are using. Understood or not? In this way we are using the names. And here if many are present, it is called polypeptide. That is nothing but protein. And here if many are present, it is called as polynucleotide. That may be either DNA or RNA based on the type of sugar present. That's it. If it is DNA, all the nucleotides will have deoxyribose. If it is RNA, all the nucleotides will have ribose sugar. That's it. In this way, you can compare these two things. Very, very important information, and every student should compulsorily understand this. Then only you can understand the NCERT lines. This is the first nucleotide, second nucleotide. So 3' OH of first one is attacking on alpha phosphate of second one. So beta and gamma phosphates are completely removed, and with the release of water molecule, here the bond will be formed, and the newly formed bond is phosphoester. Already here there is existing ester is there between 3' carbon of first and 5' carbon of second one. Two phosphoesters are there. That's why this bond is called as phosphodiester bond. That's it. This is what the information. That's it. Same is repeated for remaining other nucleotides also. Over. In this way, long molecule is produced. DNA, see, this is one strand. DNA generally present as two strands. That's why it is double stranded. RNA generally present as single strand. But please remember, even though RNA generally present as single strand, sometimes we are observing double stranded RNA also in some cases. DNA generally present as double stranded DNA, but sometimes we can see single stranded DNA also. It all depends upon organism and the type conditions. That's it. Clear. And then we are going with some important functions of this DNA and RNA. And actually this list was given in molecular base of inheritance chapter, but you need to remember this. This is very, very important information. That is the reason why here itself I am telling you. So please try to understand this information. First we will see about the function of DNA. What is the function of DNA? DNA, wherever it is present, it has only one function. What is the function of DNA? Always it acts as, always DNA acts as genetic material. It always acts as genetic material. Wherever it is present in those organisms, always it is acting as genetic material. This is having only one function, that is genetic material. It has some information inside this. With the help of this information, it will control various characters. That's why it is called as genetic material. And in which organism it will act as genetic material? Let us see. We will see some list, and then I will tell you in which organisms it will act as genetic material. You know about some viruses, and you know about kingdom Monera, kingdom Protista, kingdom fungi, kingdom Plantae, and kingdom Animalia. Please remember, these five are kingdoms. And now there is a question here. Listen the question carefully. Viruses will come under which kingdom? This is what my question is. Viruses are part of, viruses will come under which kingdom, or viruses are part of which of the following kingdoms? Now tell me, viruses are part of which kingdom? Many students are giving the answer as viruses are part of Munira. This is what the answer they are giving, but that is wrong. That answer is wrong. Please listen very carefully. Viruses are not kept under any kingdom. They are not present under any kingdom. So they are present separately. Please remember, in the kingdom Munira, what are present? Unicellular prokaryotes are present. Unicellular organisms, and all these are prokaryotes. These are prokaryotes and unicellular. Protista, eukaryotes, fungi, eukaryotes, plantae, eukaryotes, animalia, eukaryotes. And protista are unicellular eukaryotes, but fungi, plantae and animalia are multicellular. Already in the cell chapter also I have clearly explained all these points. Clear? Now let us see. Now here, kingdom Munira has prokaryotes and unicellular organisms. But what about viruses? Are viruses considered as unicellular? Unicellular means what? Single cell. Is cell present for viruses? Is the cell means plasma membrane should be present, cytoplasm should be present, ribosome should be present. These three are minimum compulsory. Are these present in the viruses? No, they are not present. So viruses do not have basic unit of life called cell. Cell is not present in viruses. So they are not coming under unicellular and multicellular. They are not part of these kingdoms. Viruses are present separately. So please observe carefully. And now what is the content we are studying? We are studying about what is the function of DNA. DNA function is genetic material. But in which organism DNA acts as genetic material? Observe this carefully. In all organisms of kingdom Munira, DNA is the genetic material. DNA is the genetic material. Protista also, DNA is the genetic material. And fungi, plantae, animalia also, DNA is the genetic material. That means all the five kingdoms have DNA as genetic material. Can you say like this or not? Yes, you can say. All the five kingdoms have DNA as the genetic material. Next point. All the prokaryotes have DNA as the genetic material. All the eukaryotes have DNA as the genetic material. Already I told you these points. Next one. All unicellular organisms have DNA as the genetic material. All multicellular organisms have DNA as the genetic material. Understood or not? And then in the viruses also, some viruses have RNA as genetic material and some viruses have DNA as genetic material. Observe carefully. Even in the viruses also, some viruses, not all viruses. Please observe carefully. For these kingdoms, all having DNA. But for the viruses, some viruses have DNA as genetic material, whereas some other viruses are having RNA as genetic material. Understood or not? So, here we are talking about DNA acting as genetic material, where in some viruses, all the five kingdoms, in some viruses plus all five kingdoms. This is what you need to write. Yes or no? Or else, some viruses plus all prokaryotes and eukaryotes, some viruses plus all unicellular and multicellular. In this way, you need to understand the concept very clearly, and this is what the one and only function of DNA. DNA always acts as genetic material. It is acting as genetic material in these organisms. Over. And then we are going with the RNA. And then we are going with functions of RNA. But RNA has various functions. What are these functions? RNA acts as genetic material. RNA acts as genetic material. Just now we studied. Where it is acting as genetic material? All viruses or some viruses? Some viruses. In some viruses, RNA can act as genetic material. So, here it is in some viruses. In some viruses, RNA acts as genetic material. Clear? And now I am asking a question that is about the human, because you can easily understand the concept. I am asking about a human. Do we have RNA? Simply I am asking that, do we have RNA? Yes or no? Is RNA present in our cells or not? Yes, we have RNA. And now if you observe, we are having both DNA and RNA also. In our cells, we are having both DNA and RNA. What is the function of DNA? Genetic material. Fine. If DNA already acting as genetic material, in the same cell RNA is also there. Now can RNA act as genetic material? If already in the same cell, if both DNA and RNA are present, definitely DNA will act as genetic material. If already DNA is acting as genetic material, can RNA act as genetic material? No. RNA cannot act as genetic material. So please observe here. In these viruses, some viruses RNA is genetic material we are saying, no? In these viruses, DNA is not present. DNA is not present. That's why their RNA is acting as genetic material. Do you able to understand this? But my question is remaining in other viruses and in these 5 kingdoms where DNA is the genetic material. In those cases, RNA is also present. Yes or no? RNA is also present. Now, if RNA is present, is it acting as genetic material? In these cases, no. In these cases, RNA is not genetic material. It is having some other functions. It is having some other functions. What are those functions? It can, in other cases, right? Not the same RNA. Genetic material RNA is different. Now we are talking about a different type of RNA. What is that one? In some cases, it can act as a messenger. Messenger. Who is called as a messenger? Messenger is the one who is carrying information from one location to other location. And that type of RNA is called mRNA. In this mRNA, M means what? Messenger. So mRNA. So RNA can act as genetic material, RNA can act as messenger, and even RNA can act as adapter. Even RNA can act as adapter. What is this adapter? tRNA. What is this T means? Transfer RNA. T means transfer RNA. Transfer RNA. Adapter means what? Link. Linker. It is linking two things. What it will link? On one side it is attached to amino acid, other side it is attached to codon present on mRNA. In the molecular base of Indian chapter, I will explain all these things with more details. Here simply just remember the examples, that's it. RNA can act as genetic material, RNA can act as messenger, RNA can act as adapter, and even RNA has a structural role. Even RNA has structural role. What is the meaning of RNA having structural role? It will form some structures. What type of structures it will form? Ribosomes it will form. For the ribosomes formation, what is the RNA that is required? rRNA is required. What is this small r means? It is ribosomal. It is ribosomal. So ribosomal RNA has a structural role because ribosomal RNA is forming some structures called ribosomes. Ribosomes, which are involved in protein synthesis. Next one. Next one. Even some type of RNA, not all types, some types of RNA have catalytic role. Some types of RNA are having catalytic role. What is the meaning of catalytic role? That means they are acting as enzymes. That is, it is acting as enzyme. So RNA acting as enzyme. Generally, who will act as enzymes? Proteins will act as enzymes. Generally, proteins will act as enzymes. But here we are talking about RNA acting as an enzyme. If RNA acts as an enzyme, it is called as ribozyme. RNA acting as enzyme is called as ribozyme. Ribo means what? Ribonucleic acid, RNA. Ribonucleic acid is RNA. Ribo is ribonucleic acid, RNA. Zyme, enzyme. So it is acting as an enzyme. So that's why it is called a ribozyme. In this way, if you see the functions of DNA, it has only one function. DNA always acts as genetic material, but RNA has various roles. In some case, it is acting as genetic material. In some case, it is acting as a messenger. In some case, as adapter. In some case, it has structural role. In some very rare conditions, even it is working like an enzyme. It is having catalytic activity. So in this way, these nucleic acids are having various functions. Understood or not? This is what the information that we are studying here. The same information I wrote here. Okay. And for this, acting as an enzyme ribozyme, in class 12th MBI chapter, you will study some examples from the NCRT. In the NCRT, they give 23 sRNA and 28 sRNA. If you can remember, remember. Or else, anyhow, I will teach you under molecular base of inheritance. There you can remember. And these two are very important examples from the NCRT. From the NCRT, and which are examples for ribozymes. RNA acting as an enzyme. This is RNA, but still it can act as an enzyme. This is also RNA, but still it can act as an enzyme. So that's why these are some important points that we need to remember. Now let us quickly read the NCRT content from class 11th biomolecules related to nucleic acids. Very less information they give. And please remember, once upon a time, structure of the DNA. Watson and Crick double helical DNA structure was given in the biomolecule chapter, but now they have deleted here because anyhow we are studying the complete structure with the details and even with the numericals also we will study. in molecular basis of inheritance. That's why here we are not studying that particular structure. So simply ignore this. In some MCQ books, unnecessarily they are asking those questions here. Simply ignore those questions in this particular chapter. Separately, we will study all the things in that molecular basis of inheritance chapter. Clear? Now let us quickly read the NCERT. Nucleic acids. The other types of macromolecules that one would find in the acid insoluble fraction of many living tissue is the nucleic acid. So in the acid insoluble fraction, we are getting total 4 types of components. Out of these 4 components, one type of component is nucleic acid. Next one, these are polynucleotides. Many nucleotides are attached to each other. That's why nucleic acids are polynucleotides. Many nucleotides are attached to each other. Together with polysaccharides and polypeptides, these comprise true macromolecular fraction of any living tissue or cell. So see, out of the 4, what are the 3 important things under the true, real macromolecules? What are these ones? Polypeptides, polysaccharides, and then nucleic acid. These are the 3. That's why nucleic acids along with polysaccharides and polypeptides, these 3 are forming the true macromolecular fraction. What is the meaning of this? Lipids are not true macromolecules. That's what they are saying indirectly. Next one. For nucleic acids, the building block is a nucleotide. That means how the nucleic acids are formed. Some repeating units are present. And what is this repeating unit is called as nucleotide. That's why nucleotide is the building block for the nucleic acid. That means many nucleotides are attached to form the nucleic acid. Next one, a nucleotide has 3 chemically distinct components. In the previous lecture itself we studied each nucleotide has 3 important components. What are these? One is called heterocyclic compound. What is this heterocyclic compound? It is nothing but nitrogenous base. So it is having heterocyclic compound also called as nitrogenous base. Next one, and the second one is monosaccharide. What is this monosaccharide? Ribose in the case of RNA, deoxyribose in the case of DNA, that is present. Pentose sugar, 5 carbon containing sugar is present. And the third one is the phosphoric acid. You can write like this or else you can write it as a phosphate, whatever it is. So these are the 3 components that are present. Next one. As you notice in figure 9.1, in the figure 9.1, in the previous lecture we studied about some structures they have given in NCERT, right? Those are the structures. There they are saying that the heterocyclic compounds, what are the heterocyclic compounds that are present in the nucleic acids? Heterocyclic compounds in nucleic acids are nothing but nitrogenous bases. Named. What are the names of this? Adenine, guanine, uracil, cytosine and thymine. And out of these 5, adenine and guanine are substituted purines. Already in the previous lecture we studied, no? There is a basic purine structure is there. In that some substituted groups will come. That's why adenine and guanine are substituted purines. While the rest means what? Remaining. What are the remaining? Uracil, cytosine and thymine, these 3. So the rest means remaining 3 are substituted pyrimidines because these are having basic pyrimidine structure. In their basic pyrimidine structure, these groups are substituted. That's why they are substituted pyrimidines. Next one. The skeletal heterocyclic ring is called purine and pyrimidine respectively. Adenine and guanine are substituted purines because they have the basic skeleton structure of purine ring. Whereas cytosine, uracil and thymine are substituted pyrimidines because they are having the basic skeletal structure of pyrimidine ring. So in this way they are having these respective rings. Next one. The sugar found in the polynucleotides is, what type of sugar is present? It may be either ribose or 2' deoxyribose. Where this ribose is present? In the RNA. Where this 2' deoxyribose is present? In the DNA. So with the for the ribose they are saying within brackets it is a monosaccharide and pentose. Why pentose? It is 5 carbon containing sugar. That's why it is pentose. Why it is monosaccharide? It cannot be further hydrolyzed. That's why it is monosaccharide. Anyhow, regarding the monosaccharides in the next coming lectures we will study slowly. Next one, a nucleic acid containing deoxyribose is called as deoxyribonucleic acid. That means deoxyribose sugar containing nucleic acid is deoxyribonucleic acid. In short, it is DNA. While that which contains the ribose sugar, that means the nucleic acid containing ribose sugar is called as ribonucleic acid. In short, it is called as RNA. That's it. In this way, very small and little information they have given in this particular biomolecules chapter. But more details once again with structures, everything we will study once again in molecular basis of inheritance. But please remember, meanwhile, you please practice these structures. Meanwhile, you please try to remember the bonds and their locations. All these things you please practice. For the practice purpose, I gave all these things so that when we are going through that molecular base of inheritance, so in single lecture I will try to complete majority of this information, because already we studied, no? So that's why I will cover major information in that particular chapter. Not only this, there are many other points they have mentioned. All those things with more details, with even with numerical practice, everything we will do in that molecular base of inheritance chapter. Clear? And with this, nucleic acids information from this particular chapter is completed. So with this lecture is completed. So in the next lecture we will go with lipids concept. Okay, we will meet in the next lecture. Thank you so much.